Hello class, this is the intro for Lab 5. It is over biological macromolecules. Basically, we're going to use 3 different tests to test milk to see what biological macromolecules are in milk. We'll always have a negative control that will be water, and then we'll have a positive control that will differ based on the test. Each test is going to use a reagent that is a color indicator. So it will change color to indicate the presence of something. So let's go over the tests, or at least the indicators. So the indicators are Lugel's iodine, and it tests for the presence of starch. And remember, starch is a polysaccharide, and polysaccharides are carbohydrates. Benedict's reagent is the next. It tests for simple sugars like glucose. Simple sugars are monosaccharides and disaccharides, and they are also carbohydrates. Then Bure's reagent is going to detect protein. It will not detect individual amino acids, which is what protein is made out of. It only detects whole protein, so amino acids that are linked together. Lugel's iodine is yellow when it's negative, and then it turns blue to black when it's positive. Benedict's reagent, let me come back to that. Bure's reagent is blue when it's negative, and it is purple when it is positive. Benedict's reagent is also blue when negative, but it is a variety of colors when it's positive. Any color change with Benedict's reagent is a positive result. So if it turns yellow or green or brown or even yellow, orange, or red, that is a positive reaction for Benedict's. You're probably going to see either an orange reaction or a green one, depending on the concentration of the sugars. You want to predict here whether milk is going to have glucose, starch, or protein. Milk is a mixture of stuff, so it might have more than one of these. I'll let you do those predictions. I don't want to lead you in any way. Just do your best guess. You can look it up if you want and be a little bit more technical. But remember, all we're testing for is simple sugars, starch, and proteins. Benedict's test for monosaccharides. You might get a weak reaction with disaccharides. Weak would be the green or brown color. And the stronger reactions are the yellow and orange and red. Your positive control will be 10% glucose. Your negative control will be water, and so there's the 10% glucose. Your glucose solution comes from your kit. The milk you have to buy on your own, and the Benedict's reagent is in your kit. You're going to place your, you're going to have 3 test tubes, and you're going to put Benedict's in all 3. You're going to put water in one, 10% glucose in another, and milk in the third. So in all these tests, number 1 is typically going to be your negative control. The middle one, number 2, will be your positive control. and the third one is your experimental value, so the milk. We don't know what's in the milk, at least not without testing. And you're going to heat the Benedict's test for around 5 minutes. Usually you get a color change before that, within like 3. And just be careful with the hot water. You'll put your results here. I'm not a huge fan of how they set up this table. You want to put the color, and then I would ignore this column and just put plus or minus below the color. So test tube 1 with water should be blue because the negative for Benedict's is blue. Test tube 2, I'm not really sure because it can be a variety of colors, but it'll probably be orange to red, and that would be a positive. And then test tube 3, I honestly don't know. So you'll just record the color there, and if it looks like the water reaction, then it's negative. If it looks like the glucose reaction, then it's positive. Our next test is testing for starch. We'll use Lugal's iodine. Again, it is yellow when negative and blue to black when positive. It's basically a dark blue when positive. Sometimes it's so dark it looks black, but it'll be a dark blue. So again, I would clean out your test tubes. On that first one, the Benedict solution may stain your test tubes, so you might want to try to clean that out as best as you can. Use soap and warm water, just like you would with your hands. And then you don't have to get every little bit of water out, but get the excess water out, and then you'll set up your next test. So test tube 1 would be water again. Test tube 2, we're testing for starch, so your positive control will be a starch solution, which is in your kit. And then test tube 3 again is milk, and you'll add iodine to all 3. And you'll record the color, and then positive or negative. So test tube 1, the water, it should be yellow-ish. So that's negative. Test tube 2, it should be blue to black, and that's positive. And test tube 3, you'll just compare it to the negative and positive controls. If it's more like the negative control, it's negative. If it's more like the positive control, it's positive. Now I should say milk is white, so it's going to affect the color some. Don't necessarily assume that's a color change. So with milk being white, it will lighten the color. But again, most of these color changes are pretty radical. The only one that's really subtle is the weak reaction on Benedict's, which is the one previous. It should be pretty obvious for starch and for protein. So our last test is for protein. It is the BIOL test. Test tube 1 again will be water. It will be your negative control. Test tube 2 is your positive control. We're testing for protein. So you'll need an egg from the store or egg white mix from the store, and that will be your positive control. If you have eggs, you will need to separate the egg white from the yolk, because the protein is in the egg white. And the specific protein in egg white is called albumin. Right there. And so that's going to be our positive control. So that'll go in test tube 2, and then of course we'll test with milk. Test tube 1, BIOL's reagent is blue, so the negative color will be blue. Test tube 2 with the albumin, it'll probably be a light purple because albumin's kind of light to clear. And then test tube 3 will be the milk, and I don't know what will happen. So again, write down the color and then put positive or negative underneath it. Ignore this last column. Alright, so here I just want you to put the colors. It'll sort of summarize your earlier tables. So put your colors here, and then when it asks biomolecule present, just record it for milk, so for test tube 3. So if test tube 3 is positive for Benedict's, put positive there. If it's negative for Benedict's, put negative there. But just put positive and negative for your milk. And then you can answer the questions at the end. And that's it for Lab 5. It should be pretty fun. It's kind of cool seeing the color changes. But that's it.